Hi, good afternoon. Thanks for coming to this section. So this talk actually is done most of the work by two of my postdocs. One is the wet lab computational people, the other is wet lab person. I think I'd like to share with you some of the ideas how to model the combination of drugs, but with mechanistic understanding. It's a new method we just started, and I appreciate any comment on that. First of all, for people to understand, drug cocktail, drug combination, a very common strategy, especially in the area in cancer. Basically, let's focus on just the simplest case about two drugs combination. And then basically you can simply have three types, same effect, just added together equal, blunt effect in the terms. That means the added together is worse than individually. And synergistic, these are most ideal cases where we look for a combination, we wish they can be synergistic. And the relationship really combined in the dosage and effect. And also the method that differentiates between additive and synergistic. So this is the area. So far, if you're in the hospital, most of the cocktail arrangement is really by trial and error. We have over 10,000 known drugs out there. And really, if you think about 2 to the power 10,000, is astronomical. So even the FDA, for the area of cancers, basically they will approve 7 combinations. Most hospitals is very ad hoc. Existing method, this is people have been working on the drug combinations for ages, actually many methods for 34 years. And all this prediction is really about what the best they give you is an index number. So-called give you some kind of index, and is it over this size bad, under this size good. So when you're synergistic at this low level, as low as possible, that's what you're getting at. But that does not give you any idea why these two drugs have synergistic. You just, by faith, just a number, you make a decision. And try it on animal, it works, and then try on human. So in this particular talk, I'd like to share with you some of the model we borrowed from other field. We call it enhanced Petri-net, used to recognize the synergistic effect of combination based on the molecular response data, and particularly treated data and after treatment data. That can be microarray. You can also be a gene expression data. It can be next generation gene sequence data. It doesn't matter. The concept is like this: before treatment, after treatment populations. For people who are familiar with Petri-net, I don't know how people are familiar with Petri-net. This is really a mathematical model advanced by person Petri-net. I think it's in the 1930s actually. Since then, has applied in many fields, even in finance, all kind of thing. In the last era, during .com, people are applying the telecommunications in the queuing theory. The idea is very simple. Basically, it's a quadruple. It's something like places and transition. And the input function make the map the places into transition into the input, and output is some sort of on the other side. Like this particular case, this token was transformed by transition 1 to page to 3, and firing of this, causing you firing and coming to the other side. At the end of it, the state changed. Initial state is 3110, and after firing of transition 1 and transition 3, there were different mapping. It's common in the communication network when you are doing the firing, when we're sending queuing theory to internet, try to figure out what's the workload in the internet. And this concept is applied for a long time. The concept we have added into is try to make it a multicolor, in the sense that we make the token in the two part. The place is called drug and gene. We're interested. So this quadruple become a 8 pairs things, right? So in this particular case, you have a places. The green one, gene token, denote gene expressions. The dark one denote a certain dosage of that gene expression. And the red one, we call it a transportation token. I can explain to you later, which is important. In this particular case, they are transferring from gene transition to this another gene. This is only gene action only. I show another one on the drug actions. And this particular one, and the difference is when this is firing the token, we leave a trace, the red one at the bottom, at the place they fire. And this is important. Why? Because really if you have a huge network, large over say, I don't know, 100 nodes. It basically gives you a trace how the token can pass through the whole pathway. And basically, by backtracking, you can actually get the drug-targeted pathway constructed dynamically from it. And also the effect of token, this is a blue and being blasted through, that means the drug has taken effect at the places. So the concept is very simple, basically just extended a Petri-net into 2 colors, and then including genes, and the other one is the drug action. And the drug action is similar thing. The only difference is now I have an input parameter placed with drugs. In this particular case, I have 6 tokens here, 5 of them are drug token. The other is red, means something is already fired beforehand, before the time equal to 1. The dosage of this place is 5 + 1 = 6. So this PG is basically the target of a drug PD1. Early on I mentioned. The firing is very simple. Now, there are many ways to optimize it. I don't want to go to mathematics, but very simple. This is basically an optimization problem. So this extended Petri-net, multicolor one, can be represented as an optimization. And you can using a simple input-output optimization to color. You can read the paper in detail, but there are many optimization methods in operation research you can apply into it. In our case, the transition algorithm is very important, the firing and basking, right? And the one we're using, very simple, we're just using the gradient, basically it's just greedy transition. Whatever is most cost effective, we just go and fire it, right? On this particular one, we call it final state oriented token transition. So transition of the drug token or gene token is determined by finding state, present state of output gene of actions. It's very simple, straightforward in the particular cases. Now, I will give you an example, right, how good of this model is. One of the examples we are using is a breast cancer area. And this particular case is a pairwise combination of 2 drugs, Gefitinib and Taxol. These are 2 standard drugs. One is target. So we're interested to find the phenotypic responses of this combination, these 2 drugs, and different dosage. So altogether you have 8 dosage pair altogether of different dosage and drug. On this particular cell line, MCF7 breast cancer cell line, we have treated and be treated. So you have molecular responses between them on this cell line, and we're using adjuvant microarray as a platform. It's particularly straightforward. And this is the result of this dose of the Gefitinib and Taxol, docetaxel. And this is the traditional method. It's by Chow in CalcuSync. He's giving you an index basically. And what he says is that anything this is actually the most popular one people used. Any index less than 1 you consider is good for synergistic. That's what the state of the art is. In our cases, we actually find out more. We will find out those pair predict by the responses, validated by literature. So in the particular case, actually, we go through browse through the literature search. We actually find out people has done on this particular dosage. A group in Japan has done in a Gefitinib on this 2 combination of these particular two areas are more interesting. Now this is only, as I said earlier on, the whole motivation of doing this is not just give you an index. The whole motivation is whether from this we can get an underlying mechanism, the pathway, why these 2 drugs add together have effect. And that's exactly what we are doing. We're trying to identify the downstream signal and related protein-protein interactions. So in this particular case, Taxol is basically focused on BLCL2, and Gefitinib is on EGFR. And basically we pull all this data from the CARE-PID biocarder and also integrate these 5 different databases of PPI in our study. We identified the target signal pathway, about 575 protein and over 900 signal interaction, and interacting with other 433. So it's very complex enough to give you some idea. And this is the overall using the PPI no direction. So the next step is really mapping from the Petri-net model we have into this molecular pathway. And this is the assigning the transition for drug and genes using the model I described earlier on. It's very straightforward. Drug target, activation, phosphorylation, inhibition, dephosphorylation, and protein-protein interaction. From this, you can generate a certain mapping. And we also do an additional experimental study. You need to find microarray for a pair combination, individual drugs with certain dosage, And that's what we did on this extender for A and B. And find out the token effect, which we calculate. And then do a subtraction, very simple, right? This will be positive if the synergistic combined effects will be bigger than the two together. That's what we wish to get on the combination drug effect. And this is the synergistic genes we identified. These are alongside with the p-value. And the idea is, as I said earlier on, this is the one we're particularly interested from our study, also confirmed with the literature. And then we zoom in more detail, and we find out more. Actually, if you go into this 8 dosage pair we conduct the experiment on, you find out it's a very interesting result. Although the two together is better than the sum, right? I mean, in the same individual. Taxol actually, in the combination treatment, has less effect. And it will be interesting. That doesn't mean that the two drugs, both of them strong, add together, they're stronger. Actually, in this case, one drug gets very strong, the other drug gets weaker, but add together, they have very, very, very good performance. And this is very consistent with the periphery phenotype study. We have the tested dosage in our experiment, original thought by the 2 dosage. And basically, the log cell is the proliferation inhibition rate significantly decreased with increasing or Gefitinib. You can see in this table, but not Taxol. Right? You can see in this area. And then we ask the question why. And this map is generated early on. I give you a set of transformation equation for the Petri-net map it to pathway. Exactly, that's why it's done. And this is the molecular signaling pathway. At this particular dosage you identify, this optimum synergistic dosage. So this one actually giving you a lot of information. In a log cell, everybody try to cater. And remember this one, early on I showed, they all targeted KRT8. This is the one, right? If this diagram, you look in more detail, the black, this is directional graph. You have blocking, you have directional, you have activation, inhibition. And in this diagram, both drugs share many gene places and exchanges. And this is a mapping. If you're using multicolor Petri-net I showed earlier on, called marking, called pi, you can mark all these transition record number in here. So if you go to further analysis, you will see some interesting phenomenon. Just observe it, but you can do computation to find out more information. But just eyeball this graph, you can see a lot of interesting things. For example, Gefitinib, EPGR has a direct effect. When this one target on this surface marker, actually you have direct effect on KRT8 directly, not for anything. But the BCL2 doesn't. If you look at this one, you have actually indirect, so direct graph to the KRT8. So Gefitinib with something called in a control theory, we call it a positive feedback loops through the many pathway. In this particular case, the RAS pathway, your EGFR, if you look at the curve, EGFR SRS1, and then HRS2, and then go down to RFA1 and then KRT8 and likewise the other path. So we have this positive feedback loops, not just a one direct hit, it's also surrounding. There are many other paths reinforce the action on the target. That's why the drug is very effective. So in a way that this feed-forward loops concept is really strengthening the transductional model. And this actually has been studied in the literature for a while. But no one really systematically tied them into the relationship using a system pathway term. And we hypothesized they form the important path to L in this particular case. And if you look at in the other ways, it's interesting. Why Taxol actually have reduced effect in this synergistic combination? Actually, Taxol actually interesting, have something that EGF doesn't have, Gefitinib doesn't have. There's something we call it control free, called negative feedback loops, in a way. Basically, if you look at this particular case, you have actually BCL2 is blocking TP53, and also you're also blocking at the same time, or blocking the SOC3 to JAK. And this is a so-called, say it's dragging their feet. So first of all, it doesn't have a direct hit on the target. Second, it's just dragging fit, reduce the impact on the target. And this gives you some idea on this graph. So, but they together work together, interesting, although one is negative. Individually, they are negative. When you put them together, actually, the role really of taxol really strengthen the positive feedback loop of gefitinib. And that's why this particular drug is so-called dominant synergistic. But also, taxol is a very common chemotherapy in drugs right now standard in the cancer area. So this is this give you a relationship, give you understanding, mechanism understanding. Besides give you an index, why this combination is good, this dosage combination is good, it also give you a reason why one is better than the other. So very briefly, what I describe is just a mathematical model which extends the Petri-net concept in the multicolor, and to predict the synergistic effect based on the treatment response data. In my case, I'm using gene expression. People think gene expression is too rough. You can use next generation gene sequencing if you have the money to run it. Now it's getting cheaper. And this really give you the importance of this model. Compare the pattern from the past and study of drug combination. Is past give you an index, doesn't give you why. This model actually give you a reason why this happened, why one thing is stronger than the others, and actually you by understanding the mechanism, actually you can predict and the effect and also design your combination strategy better. That's what we wish to do. And you can see that, of course, for computer science we all think generic, but biology is always specific. So when you want to talk about biology, it better be specific. So in this particular case, I focus on breast cancer. So they buy into it. Right now they're doing clinical trial on this combination trials on human. But this methodology, as I described here, actually, you can see can apply to other diseases. Maybe the conditions slightly different, but the concept is about the same. Right? This is really a different strategy, right? Depending how you want to attack the problem. If your user is a biologist or clinicians in a particular area, you want to focus your methodology to that disease area. But we believe this method can be extended to other diseases. We are trying right now in other disease areas like prostate, especially in the castrate-resistant prostate cancer and also lung cancer area. So finally, I'd like to thank some of my colleagues who do this work. Kai, who's helping out in our research institute, helping out the study. My colleague at Baylor, who was an NCI dedicated cancer center, Susan Fograi and Michael Ruiz provide a lot of clinical information and not basic neuroscience. Jeff Chen, who just joined Texas Medical Center from UNC as a CPRI scholar funded by Texas. Of course, it's important to acknowledge my funding agency. So after all, this is a very short talk. I'll give you a concept. To summarize, basically I provide you another look. This study depart from the past on the drug combination is you have a model. The model have using the transition tokens concept, you can actually backtrack it and provide a mechanism. What's going on in the combination? Instead of just give you a number, say these two drugs together is good. You give you a reason why it is good, and you decided from this onward which drug combination you want to use. That's all this model is about. And we will be applying in the breast cancer. Right now we're actually applying to other cancer for clinical trials combination. That's my talk. Thank you.